Hello,
We are Joshua Mathew,
Jeffrey Ho,
Joshua Cheung.
and Barber.
We are here to show you a project of the cell cycle. The cell cycle has 4 main stages.
We will overall be explaining the process of mitosis. The cell cycle is a regular pattern of growth and repair, DNA replication, and cell division.
Remember, mitosis is not the process in which the sperm cell and the egg cell is produced.
That is meiosis, which is completely different.
Interphase is when the cell enlarges to get ready to go through the process of mitosis. All the organelles also duplicate and the cytoplasm increases to get ready to divide. We can see the cell membrane and the nuclear envelope is still present.
As we can see, there are many organelles that have to be duplicated in the cell during the process of interphase. Organelles such as the mitochondria, which turns food into energy through the process of cellular respiration, the smooth endoplasmic reticulum, which is used to break down and process lipids, the rough endoplasmic reticulum, which has ribosomes on it and processes proteins to synthesize and package them throughout the cell, and ribosomes, which make proteins, are all present and duplicate during the process of interphase.
Organelles that are duplicated too, such as centrioles that shoot out spindle fibers to divide the chromosomes and organize them during mitosis. The Golgi body, which sorts and processes proteins and sends them throughout the cell as needed from the RER. Lysosomes, which cleans the cell as it breaks down many substances found throughout, and the vacuoles, which isolate harmful materials and store waste materials.
The last part of interphase is what happens inside another organelle, the nucleus. The chromatids duplicate, but they don't join as a sister chromatid. All the chromatids are floating around the nucleus ready for the process of mitosis. My name is Josh Cheung, and I will be explaining prophase. Prophase occurs after interphase and is the longest of mitosis. Centrioles start to move to the side of cells in a pair. Sister chromatids form and centromeres start to join with the sister chromatids. Also, spindle fibers start to shoot out of centrioles through the cytoplasm. Spindle fibers also go into the nucleoplasm, but not through the nucleolus. We can also see the chromosomes condensing in the nucleolus. Now, after prophase comes metaphase.
My name is Jeffrey, and I was in charge of the metaphase portion of our mitosis project. In metaphase, the spindle fibers from the centrioles that are in the cytoplasm attach to the centromeres of the newly formed homologous pairs. The sister chromatids also line up in the middle of the cell to prepare for anaphase.
Hi, my name is Josh, and I'll be explaining anaphase. Anaphase is the part of mitosis where the chromatids are pulled apart. The centromeres, which usually attach the sister chromatids together, dissolve, and the spindle fibers attach and retract to pull the chromatids to either side towards the centrioles, but never outside the nucleoplasm. That is so that when the cell splits into two, each cell has the exact same copies of DNA, creating two of the same exact cell.
My name is Barber, and I'm going to tell you about telophase. So in telophase, a lot of things happen. They include two nuclei form at opposite sides of the cell, the nuclear membrane reforms, the chromosomes unwind back to chromatins, spindle fibers dissolve, the centrioles are still present in the cytoplasm, 4 events happen in the nucleoplasm, the nuclear envelope reforms and pinches inward, but is not divided into cells, but has 2 nuclei. Cell membrane is also pinching inward, but is not divided. The centrioles are still present in the cytoplasm.
Cytokinesis is the last of the 4 stages of mitosis, where the 2 cells split off from one another. At this time, there are now 2 identical cells.
We see the amounts of all the organelles from interphase being cut in half and now placed in the 2 identical cells, as the result being that one cell divided into 2 identical copies of the original.
Now every part of the cell is visible, like the cell membrane and the nuclear envelope.
We must always remember that mitosis is used to repair and grow, and not to create different cells, but the same.